Cool. Thanks, Vince.
Yep.
Yeah, so Vince and I were just talking. I worked at Illumina before, and so every year I'd get the request to send some gear over here to the course, and myself or one of my colleagues would make sure there was always a sequencer here. And I think the last year, Dick had his MySeq here, so I think we just used that. But so it's actually kind of cool to actually be here and see what the heck actually goes on at the course. And thanks to Vince for asking me to speak here. So what I'm going to do, I'm going to take you through some slides. We can speed up or slow down as you guys have questions. And if you do have questions, please feel free to ask. It makes it more fun for me. But I'll take you through the technology, some of the data we've been able to generate, and that some of our customers in the field have generated as well. All right. So, I think when you look at short read sequencing today, it's awesome at giving you highly accurate sequence reads. As I think you guys are learning, they're typically shorter than Sanger sequencing, shorter than PacBio and some of the other long read technologies. But also, Illumina does a great job of giving you tons and tons of reads, so you can do a lot of things like counting. But one of the things that is fundamental to the way cluster gen and sequencing and ultimately the biology in the genome actually goes back to is you're taking, in this case, an analogy here where you're taking this book, you're going to dump it in your shredder, in this case your enzyme or mechanical shearing, and shred up the DNA such that it can be amplified in the cluster gen process. But then you're going to take these very short fragments and try and put the book or the page back together, which is inherently a pretty difficult proposition. And so what we do is, as Vince mentioned, is we can take long original molecules, 100 kb and greater, and put them through a process through a microfluidics. And what we're going to do is basically turn on a lot of information that you just don't get with standard Illumina sequencing today. So some of the things that I'll talk about today are haplotype phasing, so looking at maternal inheritance, being able to get large-scale structural variants, so not just the small indels you normally get from short-read sequencing, and some other stuff like new content, parts of the genome you can't really access. A lot of these things start to become tangible with our technology. All right, so how do we do that? And basically, the way that we work is we take an approach similar to what you would expect or assume with Google. So Google, you go in, you type in something in the search engine, and guess what? You get something back in nanoseconds. And what they've done is they've indexed the internet. So they know where everything is supposed to be. The moment you start typing it, it's already throwing up examples of what it thinks you're asking for. And we work in the same mode where we actually take your DNA, we're going to index it before it ever gets amplified or sequenced, so that when we go back and we see the reads that come from the technology, we can basically assign it back to that original long molecule you put into our process. And the way we're doing that is basically through genome partitioning and molecular barcoding. So what we're going to do is every fragment that results, every sequence read that you get on your Illumina sequencer is going to have a barcode embedded in it. And because we're having a very small amount of DNA in each partition, we're able to assign each one of those reads not only from the standpoint of which sample did it come from, but which molecule, original molecule did it come from. And what this allows you to do is a number of things. To be able to leverage this for whole genome or even targeted sequencing. In the same way, it actually leverages a lot of the workflow that you guys are probably learning in this course. And most importantly, it leverages existing Illumina infrastructure, so there's no need to buy a new sequencer to get all this information. We're not the first ones to think of this, however. There's been technologies, academic and even commercial, that have tried to do this. But one of the big things that we've solved that some of the other technologies haven't is that while the ability to partition or append in barcodes is rather limited based on the lack of microfluidic approaches. So some of the technologies that are out there, they're limited to plates, so you have to put each well as a partition. Each partition gets one barcode, but the plate format actually dictates the barcode or the partition number, and typically requires a lot more DNA. So the approach that we've taken actually can scale this massively, so greater than 100,000 partitions is capable in our instrument, and we're drawing from a barcode pool of greater than 750,000, and all the while it's actually running the whole assay off a nanogram of DNA input. So it's very efficient in how it works.
No way.
Yes, way. So here's the instrument here. It's pretty small, so Vince can tell you about the one that they have there at WashU, but it's about the footprint of my computer here, about 13 pounds. And basically, what the kind of two most interesting parts of the system are the microfluidic chip, which is right here. So we can load up 8 samples worth of material in the system. And this right here is the gel bead. So each one of the wells inside this strip tube contains 750,000 barcodes that are deposited into the system here. So there's only 3 positions. We'll show you those in a second. But the processing of or loading up the chip takes about 5 minutes. Load that in an instrument. Instrument's going to run for about 5 minutes. In that time, you've done about 1.5 million pipette steps. So it's a very fast and efficient way to partition the DNA. All right, so let's take you through the technology and how we actually make this all work. Kind of the unit of operation to really kind of think about is what we call a gel bead. There's a lot of cool properties about the gel bead, but for the purposes of simplicity, you can think about it as a very efficient oligo delivery system. So what we've done is we've built kind of a hydrogel-like structure, so it's empty on the inside except some scaffolding where you can hang more oligos. And what we do is we basically hang tens of millions of the same oligo, which is kind of represented by this little fuzzy surface here. But each one of the oligos basically has machinery that's going to interact with the Illumina sequencing process. So essentially the complementary oligos that are represented here, this P5 sequence, there's a complementary sequence on your flow cell.
Where's the P5 sequence at? On the fourth adapter? That's right, right?
Yes. And then the barcode structure here. So this is, and I'll show you an example of what this ultimately looks like. You guys are probably still learning this, but what we've done is we've taken a barcode position that's normally there from Illumina, and we've hijacked that to essentially index the genome. And so what that allows us to do is basically barcode every one of the reads, resulting reads that come from that process through this 14 base pair barcode. You also have your R1 sequencing primer, and then the NMER, which is the kind of functional end of the oligo that's going to interact with the genome in the presence of our biochemistry. And so we make all of these in our factory in Pleasanton. These are all vanilla beads that start out in life, and we functionalize them with each one of the 750,000 oligos that we produce, put those on, and then go ahead and drop those in a tube and send that off to the customer.
So IDT loves you guys?
We aren't buying 750,000 oligos. We wouldn't have too much of a business if that was the case. So how does it work? So you saw the microfluidic chip a couple slides ago, but what you're looking at here is a cross-section of the inputs into the microfluidic chip. So what you're going to do as you're loading up the chip structure, you're going to load in your gel bead. So it's just going to take from that 8-well strip tube, load that into one of the positions. And what's going to happen is the other 2 positions are filled with your DNA and a partitioning well. So physically what will happen is a manifold is going to come down. Pressure is going to force the gel beads from left to right. So from here to here. And what's going to happen in this first cross, you can kind of think about it as a kind of a highway on-ramp. Cars come on, the DNA is kind of sitting around side the gel bead as it's moving through the channel. And once it hits the second partition, or second cross, oil is going to come in and basically envelop one gel bead, which has tens of millions of the same oligo, and about 100 or so DNA fragments. So you can think about, you've got about 3 to 5 femtograms worth of DNA in each one of these partitions. Okay? And what's going to happen, they're going to keep going through, they're going to collect in the outlet well of the chip, and it's going to keep going and going until about 5 minutes when all the reaction is done. So what do they look like? You can see that right here. The kind of dark outer ring represents the boundary of the partition itself. And if you can see it right here, there's kind of this whiter inner ring which represents the boundary of the gel bead. And so basically you can see that 2 things about this picture. One, that the gems themselves, these droplets, are very monodisperse in their formation, and the gel beads we build are also very monodisperse in how they're built and delivered. Now, not all of these droplets are filled. I think that one right there is empty. There's maybe one other empty one in this image. But what we've been able to do is actually tune the rate of the two crosses. We call them Z1 and Z2, but basically we can tune the speed by which they kind of interact with one another such that we can get a very high fill rate, greater than 90% through the process. Now, one thing to point out too is that we're putting in really long molecules in the system. What we've done is we've actually screened a number of available DNA extraction kits. We certainly didn't want to make people go through heroic efforts like gel plugs or other kinds of crazy stuff to get long DNA. So we screened a number of kits. The one kit that we've seen the most success with is the QIAGEN MAGATRACT, which is a bead-based extraction kit, and that gives us very long pieces of DNA from that process. It's fast and easy. There's no columns, none of that crazy stuff. And here's an example of a length distribution of the molecules that we've seen go through our system. So you can see out here there's a kind of a small fraction, but a fraction nonetheless, at 450 kilobases. And one of the big challenges we have is that just to be able to measure really long pieces of DNA requires things like pulse field gel boxes or other things. So it's just the ability to measure the DNA that we're putting through the system becomes challenging in its own right. But what we have seen when we can accurately measure it with pulse field gel is that we're putting very long pieces of DNA through the process and they're working just fine. All right, so let's kind of go through the biochemistry of actually what's going on now. So I mentioned the enmer on the end of those gel beads. The one thing I didn't mention is that the gel beads themselves actually dissolve. So they're not a solid bead. Once the gel bead actually gets in the droplet, there's a biochemical process going on. So what's happening is that the gel bead is dissolving and releasing all of those oligos into solution. And so the reason we have so many oligos in solution is we want a high molar excess relative to the DNA that's in the droplet. And what's going to happen in each one of these droplets is that what we do is we take the product from our instrument, we go ahead and load that on a thermocycler, and with the oligos, the DNA, and the enzyme that you put in also through the process, in the process of thermocycling, what's going to happen is that enmer, which is now free in the solution, is going to come down and it's going to hit a part of the genome, right? So there's a random sequence at the end here. It's going to hit, and it's going to do a primer extension assay across the length of this fragment. So you're going to get a fragment that essentially has, you can think about it as 1/2 of a library molecule right here, and you end up with a product where in this case it's about 1,000 to 1,500 base pairs. So it's starting to get on the order of what's compatible with cluster generation. It's a little too big in this form right here, but at least it's on the order of what is compatible relative to, let's say, a 100 kb fragment that you might be starting from. And so we're going to basically sample lots and lots of these parts across any one of these single molecules, and we're going to go ahead and collect that. And then what we'll do is we'll break the emulsion. We'll just hit it with a single reagent. It takes everything back to homogeneous aqueous phase. We do a spry cleanup to remove any of the unincorporated primers. And Vince, have they already gone through library prep already?
We all know about spray.
Okay, cool. So basically the process that you see down here should be the process that you guys know or are learning here, which is basically to shear. In this case, we use a Covaris, which is a mechanical shearing device, to get it down to the appropriate size range for cluster generation. We're going to end repair, we're going to A-tail, and we're going to ligate on the other side of that library molecule. So your P7, your read2 sequencing primer, and at this point, you can also do a sample index PCR. So this would be how you would make sure you could batch lots of samples and still be able to discriminate those informatically, but get the most cost-effective use of your sequencer. And then from there, you have a library that is compatible with Illumina, and it looks pretty much like you would expect an Illumina library construct to look. And as I was mentioning, where this is normally taken up as a position to do sample batching or sample multiplexing, we've hijacked that to index the specific genomic DNA fragments before they get sequenced, and then all we do is we still give you the same level of plexy, but we've just relegated all of that plexity here in the read2 index position. Does that make sense? Question?
So for your random endments, how do you make sure that all of them don't bind kind of close to each other? How do you get them so nicely spaced out?
I'll show you. Actually, I have a picture where you're going to see that they're randomly spaced. So in some cases, they will bunch up. In other cases, there'll be long stretches where there isn't any reads. So the goal ultimately is that we aren't trying to carpet bomb, let's say, one fragment and fully reconstruct it, but rather give you long-range information with kind of an economy of sequence reads. So I have a picture, it'll probably make it a little bit more clear. Yeah, good.
Yeah, I just wondered about the gem code barcode. So which read when you do Illumina is going to be actually reading the barcode? Because I see read 1, read 2 of the sample.
Yeah, this is going to be your index one position.
Other index one.
Yeah. Yeah, I5 would be in that vernacular. So the process here, basically you're taking an anagram of input, putting it into the system, and one thing we can do, we can either, and I'll show you a couple of data examples, where we can do either whole genome or whole exome through this process. So what you build is a library. What you choose to do with it, whether it's genome, exome, or even some sort of custom hide capture is really up to you, save for the shearing step. And then basically this is of course compatible with the Illumina sequencing system, and then I'll kind of take you through some of the software that we've also built to address the data type that we produce. So one of the things, Elaine could speak to this, but from the standpoint, having worked at a number of the sequencing companies, a lot of times people choose to keep the data type closed in for their own reasons. And so what I was pretty happy about is when I joined 10X, to say whether we should keep it closed source or open source, it was an easy decision to go open source. So what we've tried to do is actually build everything such that people like yourselves can actually build tools that will leverage the data type that we produce, and rather than try and keep it all in-house, we don't have a big enough team to do all that stuff, so it's more advantageous for us to actually have people out in the world actually using the data type and figuring out what they can get from what we've built here. And so that's just one aspect. The other aspect is we didn't want to build things that already existed. So if there was something good in the world like BWA for your alignment step, we were more than happy to use it. For variant calling, GATK or Freebase, which are obviously freely available and widely used, we've leveraged that. Although I'll talk in just a second, one of the things that we are developing and will be launching is a barcode-based aligner. So this will start to take advantage of the barcodes where these tools, as they were not intended or built with us in mind, don't. So we're starting to kind of add on tools that will allow people to leverage more and more of what our data provides. But what we did build are things where it didn't exist or we needed to. In this case, we have tools to help with the haplotype assignment, structural variant calling, and ultimately a way to visualize the data. So something like IGV is not haplotype aware. So just to make sure it's clear, when you look at a sequence read from Illumina, you've got mom and dad that are each contributing, but the way Illumina works is there's no way to disambiguate which allele is being called, right? Is it from mom or from dad? It's all superimposed on one another. And that's what IGV shows you. So what we had to build is a haplotype-aware browser so we can actually say, a variant, which allele is it actually coming from. So that's another piece of tool that we had to build there. There's lots of information. I won't get into the details because I don't know all the details myself. But we've got a fairly intricate set of tools that live underneath the hood. And you can certainly go here to software@10xgenomics.com. And basically we have a whole bunch of, you can get all of the data that we have available. We have a number of data sets. There's a lot more details about how our software works. So if you're interested, definitely encourage you to go take a look at that. Now, one of the things that we're going to be launching next month are a couple of advances which I think will be quite useful. We've had a number of requests for people that want to do things for which our original software set wasn't necessarily built to do. And so we've had a number of requests. We actually have something called the 10X Zoo for all the different animals and species and things that people have asked us that they would like to do with our technology. And so what we're developing or going to launch is something called a basic pipeline, which is basically rather than taking a reference, there's no reference used at all. All we're going to provide you is barcoded reads, and then you would be free to do, let's say, de novo assembly or other kinds of things with kind of a very generic data type. But then we've also had requests where people come in with very different references than, let's say, just HG19. And so we've also put together something called a liner where you can put in your own reference not just HG19, but you can do other things with it that will allow people to do their own specific variant calling against that. But what I'm really excited about with this release is this part. So if you think about when people do, let's say, a whole genome sample today, the genome is very complex, and it's kind of like the last mile is always the hardest to get, right? So when you do a genome today, you're getting something like 92, 93% of the genome. And the reason you're not getting the full 100% is that those short reads that Illumina has, it's harder and harder to uniquely align those because of the repetitive elements in the genome. And so what we are launching is something we're calling 1.2, but what it does is it basically takes an iterative approach to aligning reads. And I have a couple more slides, I'll talk about it. But what we've seen is that we can start to go into parts of the genome that are a little bit dark to Illumina in the sense that it's hard to uniquely map or align a read. And we can do an iterative process where we anchor a read that's highly confident in terms of its map score. And then we basically reduce the complexity of all the other reads that didn't align, and we use that to kind of iteratively walk our way into these more difficult parts of the genome. And so we're seeing 100,000 extra new SNPs that from everything we can see doing some validation look to be legitimate SNPs. So I think it should start to rescue several percent of the genome just by using this variant call, or new variant aligner that we're going to be launching next month. All right, so your question earlier about linked reads. This is what the data looks like. And so what you can see, where's my cursor? There it is. This is a 100 kb stretch in NA12878. And each one of these colors represents a unique single molecule that you put into the system. The dots represent an Illumina read pair, right? So both the two ends of that same fragment. And what you can see is that, to your question earlier about, how do they land? They do land randomly, right? So you can see on this purple one right here, right? There's a stretch where there isn't any reads. And this, for this particular molecule, there isn't. But if you look down, then you still get the same depth of coverage you would normally expect to see with Illumina, right? So it doesn't require us to be exhaustive on any one molecule because you still have the depth of coverage for any specific loci that you'd be wanting to interrogate. Another way to think about this is if you didn't have us, each one of these read pairs, you'd still align them based on the reference, right? Can I uniquely align this and say, yep, this is exactly where it goes? But you'd have no context of anything up or downstream of that other than what the reference provides you. And so by the use of the linked reads, we're able to say not only does this map here because it should go here based on the reference, but now this read, and all of these reads also belong not only in their correct positions based on the alignment, but they go on the same molecule because all of those reads share the same barcode that was present back in the droplet formation step.
Okay, so can I just ask a basic question? So back to so I understand exactly the bead. So each bead is only one barcode. That's
Correct.
Barcode, right?
Correct.
And there's more than one. There's 100,000. I mean, there will be more than one bead with a given barcode in the pool of beads.
Correct.
But one thing I guess. So obviously two different molecules will attach to the bead, right?
Yeah, you know what, I think
But I mean, is it just an odds thing that the odds are two molecules that are different attached to the same bead that they're probably not like the exact same region?
Yeah, so you know what, I was focusing on a bunch of parts here. I forgot to talk about a very key point, which I think you're highlighting here, which is we're running in a haplotype limiting dilution, right? So basically, you're absolutely right. We're taking a really small amount of DNA, and think about this as like 100,000 well plate. And you're taking a nanogram and you're aliquoting across all these 100,000 partitions. And so when you have a few femtograms worth of DNA, the likelihood that you see two fragments sharing the same loci in the same partition is really, really low, right? And so basically, right now, if you were to go and load 100 times more DNA, the thing you're talking about would absolutely happen, and your ability to call would be severely compromised, if not completely broken. So yes, that is a key aspect of the technology, is to put in a very small amount of DNA, because from there, there's really two ways. You can either do more partitions to further dilute out the sample, or you put even less DNA in, right? But we've tried to find a kind of an optimum balance where the amount of DNA is kind of actually feasible to actually put into the system, quantitate and all of that stuff, and that there's enough partitions to spread that across. But sorry, I kind of glossed over this slide, but that is absolutely critical to how our technology works here. So the other thing that's interesting about the technology, though, is, if you think about some of the other long-read technologies, let's say you just cared about exonic regions, right? So these blue bars here, and you had a true contiguous long-read technology. There's no way for you to say, only give me this. What's going to happen is a long read is just going to keep running through all of this intronic space. And so while you could do, let's say, a bait capture with PacBio or something like that, you're going to read through all of these regions you don't care about. The only way PacBio would work the best would be to take a very long contiguous fragment and pull that down and sequence it, which is a different way to use it. But for us, it allows us to kind of tap in to kind of what people do a lot today, which is these kind of custom hide captures or exomes, where you're just looking at a small amount of the genomic regions of interest and being able to sequence those very economically. And what you can see is, even though it's this whole genome up here, you can still see that the reads are basically aligned against these exonic regions that correspond to the baits that were put into the set from any of your favorite bait capture providers. So it's a nice way to get kind of long-range information from a kind of a data type that people don't normally associate with long-range information. And then one thing just to point out here is that the workflow is pretty much the same. The only difference is, for us, if you're going to do whole genome, you'd shear to kind of a higher size range on the Covaris. And if you're doing exome, just because some of the things people care about is maximizing the reads on target, you don't want to sequence the ends which are outside of the targeted region. You can do it, but you're just wasting money. So this is a way to make sure that the fragments that are actually put through a bait capture are kind of size appropriate for the Hive baits that are actually in that kit. So in terms of what do we get from it? We get whole genome phasing, where we can get very long stretches of the genome that are in the same phase block. So we see pretty routinely, 10 megs and above as an N50. And then for us, we also, right now we're tapped out at about 40 megabases as a phase block based on some software limitations we currently have. But the nice thing about this is that we're doing all the haplotype phasing from the underlying biology. We aren't kind of imputing to basically rescue and put things back in from this approach. And same thing, we can do the same on the exome. And the only difference that we see in the exome is that the way that the baits are spaced aren't necessarily the most advantageous for us, because you end up in regions where the size of the DNA relative to the spacing of exon baits can be larger, and therefore it's harder to join those two things. So that's why you see a slightly lower performance in terms of phasing. But as I'll show you in a second, we do have some data where we've actually spiked in baits and been able to rescue things we know are biologically present, just not present in a bait set. All right, so a couple of, I think from here on out, I'll just show you data at this point. But let me ask, is there any questions about the technology before I kind of jump into data that I didn't get to, or if you guys still have burning?
I'm curious about, so it seems you would also be able to, you talk about long reads and phasing of haplotypes, but what about, and maybe you're going to talk about this, accurate detection of somatic variants in cancer? Is that something?
Yeah, it's definitely something that we haven't focused on it yet, but we do have customers. I do have a couple of examples of people that are using this in cancer variant calling, and I think some of the stuff about with our 1.2 release is also going to be quite exciting for people that want to do, because you're going to be able to read into hard parts of the genome and be able to get regions that you just can't get today. But yes, you can do cancer sequencing. I'll show you a couple examples of that as well. And actually, this is a cancer sample right here, where we took a cancer cell line, H2228, and what we were specifically looking for was the EML4 ALK gene fusion, which is pretty well known. And you wouldn't be able to see this in a standard exome because this is an intronically mediated gene fusion, right? So the thing that you need, you could see this pretty easily in a genome, but you wouldn't expect to see it in the exome given the distance, not only between the two parts, but then also where the baits land relative to this. And so what you can see is basically what we're counting up here. You can kind of read along this edge where we've basically seen, here's the ALK on the y-axis, and then these exons in EML4. And you can see basically we're just counting up the number of times we see a barcode that basically says these two parts of the genome are proximal to one another. And so if it's from the reference and you'd expect it to be there, great, no problem. But in this case, where you wouldn't expect it, you'd see this read pileup evidenced here by this kind of checkerboard pattern, and you could say, hey, I'm actually seeing evidence of this kind of gene fusion. And when we looked at this in a standard control, NA12878, you don't see any evidence of this in there. The other thing that the technology does, I think gets a little bit to what you're asking, is around the ability to call variants in cancer samples. So one of the things that is really challenging is when you think about there's really two problems, I would say. One is that the input load a lot of times available for cancer samples isn't always that high, right? And then the second thing is as you drop the input load, which you can kind of see here, for example, Agilent recommends 200 nanograms into library prep. But as you kind of decrease the amount of DNA going in, your PCR duplication rate, which is making essentially copies of copies or junk, goes up pretty high. And the regime we're operating in is much, much lower, where we are in the range of anywhere from 1 to, I've seen maybe 4% PCR duplication rate with us at a nanogram. And so you can get very, very highly complex libraries you can sequence deeply, which would be really important, especially if you're trying to get very low minor allele fraction variants in a particular sample. And so another way to kind of look at that and say, well, how do we do? This is data from Agilent exome where we did the recommended amount, and we get a certain amount of PPV and sensitivity at 200 nanograms, and we're getting basically equivalent performance at 1 nanogram. And if we titrate down to 25 nanograms with a standard library, you can see that the hit you take because of the PCR duplication rate. So it gives you a lot of power to sequence more deeply and make sure that you're sequencing all the unique and informative molecules in that process. All right, so just a quick plug here. We do have data that's available online. So if you go here, again, is software.10xgenomics.com. We actually have samples that we've made available. You can go download these, go to town on them. And then we also have an online version of our Loop browser. So you can actually go in and see parts of the genome with our haplotype browser through this webpage here. All right. So as part of the data release we did for the NIST samples, we sequenced basically an Ashkenazium trio as well as NA12878. And you can see the phasing performance there. And this is kind of what I was talking about earlier, that our longest phase block right now is pegged out at 40 megabases. We think this can go higher. It's just based on the current way we've architected our software. So once we change a few things in there, this should jump up. But you can see you're also phasing a high fraction of the SNPs that are present in each one of these samples. So it gives you pretty nice information about the correct haplotype assignment. And you can also see here the average DNA length that went into the process, ranging from just over 100 kb to over 150 kb. But what's kind of cool is that you can actually start to basically phase an entire chromosome arm with the technology. So thinking about something that's 40 megabases, in this case 39.1, and you can basically say that all of these SNPs are correct with respect to the inheritance pattern there. So it's kind of a cool way to start looking at the genome. Again, the linked read plot down here. Now, one of the things we did as part of this release is we don't use imputation to make any of the calls, but because this is part of a well-known trio, we could go back and confirm the inheritance pattern based on the availability of the sequence data. And so what we were able to show in this couple of examples where NA12878 is not a very biologically interesting sample, not much is going on, but what we were able to go in and we could find some of these areas where there are kind of genotypes you wouldn't expect to see with standard short read sequencing, but we can start to fish out because of our unique property with our technology. And things that are kind of cis or trans with one another, you wouldn't expect to see. You can start to fish those out pretty easily with our data type. That one, yeah, you can see that the length of the gene, 106. Phase block where this is located is 29 megabases. And then another example where basically you have 2 mutants that are cis with respect to each other, and then one is in trans, so this would be a very hard mutation to call with standard short read sequencing. Now, the other thing we can do, of course, is call structural variants. So in this, this is one of the Ashkenazi samples where we're able to sequence, again, show everything compared back to the inheritance pattern to know that we did get the correct call, but we can see very clear evidence of, basically a deletion that's coming in from the father and then carried on to the child, but which isn't present in the mother. So, and here's another way to look at that. It's when you look at it in the linked read view, you can very clearly see that this allele, no deletion present from the mother, but then obviously through inheritance, the child inherited the one deleted copy from the father, and then the mother had a perfectly working copy. So it gives us a really nice way to boost your signal to noise when you're starting to call things, because you can make the correct haplotype assignment, and then from there do a much better job of calling SNPs or other kinds of mutations. The other thing that we can also start to see are retrotransposon events. And so if you think about each one of the transcripts right here, and what you can start to see is each one of those transcripts kind of gets its own linked read plot, so to speak. But the nice thing is you can actually see the transcript, which was actually where the variant was retrotransposed in there. So it's kind of a cool way to start to see some of these things that are biologically active and relevant, but you wouldn't be able to normally see those with standard short read sequencing.
So to make these graphs, do you have to kind of know your areas of interest, or does it just make these for anything that it thinks you'll find?
That's a really good question. So, so what's happening, you don't see just the way we've cut off the graph, but our software will make all these calls. And so what you're going to do is basically there's a whole row on the left-hand side, and you can just click. And once you click on it, because it's making all these calls based on quality scores, and all you're doing, this is called the matrix view, and it's just taking you to those graphs to say, here's an event that we called, here's some quality score, here's where it's happening in the genome, and then here's the barcode evidence to support that claim. Good question, though. All right, so last couple of slides. This is kind of what I was mentioning earlier. So we've been working with Serafim Batsoglu, who's a computer scientist at Stanford, and he basically came up with this mathematical approach he was calling RFA. And basically what he showed in some of his own work is that he was able to rescue a whole lot of SNPs using this approach that he's developed. And basically, it kind of gives you a little bit more detail where he basically takes the reads, he aligns them once, and then from there you can kind of do this kind of iterative process where you can essentially walk into these more difficult parts of the genome, where if you think about, let's say, a standard short read, you're going to kind of live and die by the ability for the aligner to uniquely place that read. So if it's in a region of high homology, GC, or some of these other things that can confound standard short read Illumina sequencing, that's it, right? It either aligns or it doesn't, or it aligns with some level of poor quality. But what we can do here in this approach is basically align a read confidently, and then from there kind of reduce the complexity space to something a little bit more manageable for the aligner, and also use the local context of that barcode to then go back and say, well, now can I place this read? Can I place this read? And this is kind of an example of what you can get with that. So in this case, PacBio, which is obviously a very long read technology, they were able to go into region and say, hey, great, we got great coverage in this particular part of the genome. We've got variants that we can call pretty nicely. And the problem with poor Illumina is that those short reads in that region of high homology just don't align very well or at all. And so by using the same approach from RFA with our technology, you can start to basically place those reads and recover that variant that would have otherwise been lost with standard short read sequencing. So it's kind of a nice way to just, from a data perspective, get a lot more of the genome back than you would otherwise get with standard short read Illumina sequencing. And we've got a number of examples that we're starting to fish out. So I'm pretty excited about this improvement with the technology. And this is another, it should be M, damn it. This is another example of a structural variant where we worked with a researcher at Dana-Farber, where he's very interested in prostate cancer. So we sourced the VCAP, which is a prostate cancer cell line, and we really wanted to see, could we see TMPRSS2 ERG? And lo and behold, we can see it just fine in whole genome. This is a gene fusion that's on chromosome 21. So this is kind of your question. This would be an example of a call that's made on the left-hand side. So you can see, here's the quality, where is it occurring, what's the position, which genes are involved, and what's the distance between those two things that they're acting, right? And so you could go pull up the matrix view and say, boom, there it is. So we know it's biologically there. However, when we went and did exome sequencing, and again, this goes back to the spacing of the baits relative to the size of the DNA, we saw some signal for it right here, but not a lot. And so what we did is we actually went in, synthesized baits, spiked them in. So these are intronic baits that are spaced every 2 kb, and you could start to see much more clear signal for this, this particular gene fusion. So we were pretty excited about that. And then we got the data and we started looking through it, and great, we saw the gene fusion we wanted, but guess what? There's a whole bunch of other stuff that's actually going on. So all these other genes are finding other partners, and each one of these gene pairings basically comes with its own unique signature, which is evidenced by all these barcoded reads. And so we were like, wow, this is pretty crazy. And basically what he had shown in a paper from 2012 is that, basically you have this chromoplex here, shattering of the genome, basically has this kind of near catastrophic event, kind of jumbles all together and then goes back together in kind of different and weird ways. And so essentially, what was kind of interrogated through a lot of pain and effort in this paper, we could essentially recapitulate and add some new information to which genes were being involved based on the same cancer cell line. So it's kind of a cool way to think about what you can do, in this case, to get from the back of an exome. A couple other examples, and I think I'm almost done here. We've been working with Hanli Ji, who's an oncologist and works at the Stanford Genome Technology Center. And so he's got a number of really interesting sample sets that he draws from his practice. And in this case, what he was doing was looking at a colorectal cancer sample. And what you're looking at here is basically what we call the reference line. But you can see that we're basically just looking at the same part of the genome kind of back on itself. And this line, if everything's normal, should be a nice diagonal across that region. However, because in the normal sample, this is indeed the case, but we look at the same part of the genome, in this case in the tumor sample, you start to see basically an amplification event, and that's kind of measured by the distance from the middle of this line outward. And basically it shows that there's essentially a tandem duplication event that's going on in the genome based on this kind of broadening of the barcoded reads that you see here in this particular graph. So this is a pretty cool example and something that he had seen by other technologies. Yeah?
What is even in the normal, there's some regions like almost the end of Cat6A, there's a white cross in there. What is that? Does that mean that that's I'm thinking that's an example where there weren't too many molecules that bridged across that point, right? Yeah, you could, does it mean anything at all? I just noticed that pattern.
No, not really. There's going to be regions where you don't see like a barcoded read, that it will kind of dip down, but as you can see, it's...
That's aligned to the reference too, right?
Yeah, this is aligned to the reference as well. So it could also be even problems with the reference at that point. We actually kind of run into that a lot, where the reference is actually wrong relative to some of the barcode evidence that we have. So. And the last one, this is a more recent example where he's got a diffuse gastric sample, gastric cancer sample and was able to evidence an amplification in this particular gene. But I think to your question earlier about cancer, what was interesting about this example, I believe the tumor purity here was something like 25%. So basically you're able to start to pick up things at kind of lower and lower frequencies, because you think about, I have to call something that's very rare in the population, and then I've got this mixture of, all of these things. So again, the ability to have low amount of DNA, spread that across, have more partitions, gives you a lot more power to start to pick up some of these somatic events here. So with that, just want to say thanks, and if there's any questions I can answer for you that I haven't been able to get through in the talk, be happy to address those now. Otherwise, thanks. Yeah, go ahead.
Again, do you have a sense, have you guys done dilution series, you know what the limited detection of a single nucleotide variant is, or, you said that sample was 25%.
Yeah.
Can you go down to 5%?
I think the answer, well, the honest answer is we don't know we haven't pushed it that far, but we do have, I know some of our customers, they are trying to push this probably faster than we are, where, for example, one of the things you can do is I was alluding to this earlier is, if you're trying to get to really, really low events, you could basically take advantage of the real estate on one of the chips. And so each one of those channels in the chip is going to produce 100,000 partitions. And so what some of the customers are doing is they're taking a sample, and it's the same sample. All they're going to do is process it the same way, but they're going to have a different sample index. So all of those things came from the same sample, but you can uniquely assign them. What that's going to buy you is you're going to have more partitions to spread out that DNA. So instead of 1 anagram across 100,000, it's essentially taking 1 anagram across, let's say, 800,000 partitions. So that's one way where you can start to make sure you're getting further and further dilution of the DNA across all of those partitions, which would go really, really well towards getting very dilute samples and going down to those lower tumor purity examples there. So that's one way to do it. The other way, of course, is you can sequence more deeply. So there's a number of ways to play it out, but that's kind of like the simplest way that a couple of our customers are actually trying this right now.
We've done that in the droplet PCR, just multiple samples, the same sample,
Yeah.
reaction or something with lower VAS.
Yeah. So, but yeah, Han Li's example was pretty nice because he's pushed it much further than we've tried internally. Most of the samples we've done, just for ease of accessing, are cancer cell lines, and the purity in there is obviously pretty darn good. So we haven't really kind of faced a lot of the challenges that people in the real world face, which is I've got something that's very, very dilute with respect to the thing I'm interested in, and but yeah, we do have some customers that are really starting to explore that. And I think there's a few things we can do that will help to improve that as well that we'll probably be talking about next year. So, yeah.
What's the lowest amount of starting material that you've done with that?
Lowest amount? Basically, I think we've done about 250 picograms. About, I would say 300 is probably like a reasonable floor. You probably wouldn't want to go below that, but yeah, we did about 250. We had some, we did a collaboration with Jay Shanduri at University of Washington. He had these, horribly degraded buccal swabs, and they, basically just want to see, kind of a little bit along the lines of your question, which is, can I just call variants? Like, forget long-range information. Can I get SNPs out of this? And these things are so horribly degraded, but because of the PCR duplication rate is so low, and even though you had a low amount of starting material, we could still build very complex libraries you could call SNPs from. So.
How do you get this thing where there's so few you start talking about this limiting pollution, but
Yep.
you also said 90% of the beads have, right? I would think that that in order to get to 90% of the beads being occupied, you'd have a lot of is that because of the microfluidics?
Yeah, that's right. So one of the things, I kind of verbally said it, but it's probably easier with a picture, but so right here, basically what we can do is the rate of incoming gel beads into this cross, and then the ejection rate into the second one, we can tune that speed. And so that speed, from here to here, we've optimized that so that basically all the 90% is essentially saying that of all the droplets that get formed, 90% of them have a single gel bead. Now, you're going to get partitions that have 2 or none or something like that, but the likelihood that you get kind of these other non-ideal implementations is quite low, because obviously, if this wasn't efficient, we'd have to increase the DNA load, which would break the haplotype limiting dilution. So you need that to be very precise, and that's what we've done with kind of the geometry, the pressures, and all these other things we've had to optimize in the microfluidic setup. So.
Anything else? All right, thanks, Rob.
Thanks. Thanks to you guys for making it interesting too. So.